Hi everyone. I hope I'm audible to you. Can someone just maybe unmute yourself or write in the chat if I'm audible? Am I audible? If you don't want to unmute yourself, you can write down in the chat.
Audible, ma'am.
All right. Thank you so much. Okay. So maybe I'll wait for a few more minutes till 6:05, and then we can start. So that if there are any others who want to join, we can join. All right then, let's start for today. Hi, I'm Anaka and I'm a research scholar at IITB and I'll be having these about four or five of the problem solving sessions for this course, which is Plant Development Biology. Um, we will be having four courses since it's, 4 weeks where we'll have this problem solving since it's a 4 week course, and then I think one extra. And whenever the due dates for the assignment get over, we can discuss them. Till that time, we'll be discussing problems related to the week that you have studied, but not the assignments. Okay. So once the assignment due date is over, we can discuss that also if you have any doubts in them. So let's start with the questions for today. I think I have about 14 or questions. So anytime you have a doubt, you can stop me and ask. And any answer for the questions that you want to give, you can either write it in the chat box or you can unmute yourself and talk to. Okay. So I hope that was clear. So welcome everyone and let's start. So the first few questions are from the 0 week assignment, and this is just in case you have a problem with any of the questions or you don't understand it. So just to clarify it, we'll be clearing this question itself too. Okay. So feel free to write down the answer that you think is correct in the chat box, and we'll see, right? So, which the question is, which of the following is the male reproductive organ in the flowering plant? And there are four options: calyx, gynoecium, androecium, and corolla. So, let let me just see the chart. So, Jyoti says three, and that is the correct answer for this question. The answer is androecium. So, this portion of the flower, okay, which has the stamen and the anther, is called androecium, and that is the male reproductive organ in the flowering plant. So great for you guys. So since you don't, I'm assuming no one has a doubt in it. Just stop me if you have any doubts in the answers, okay, or in the question itself. Until you tell me, I'll keep moving on to the next question. Okay. So question number two is the energy currency for any living cell. RNA, DNA, ATP, and AMP. Yes, yes. So everyone is saying ATP, and that is the correct answer. So from the diagram that I've given you, you can see that ATP will be converted to ADP because one inorganic phosphate will be removed, and this reaction releases energy. So ATP is the answer. RNA and DNA are nucleic acids, as you know, and AMP is also not in the form. AMP is not a energy currency because it doesn't have a free inorganic phosphate that can be released. Okay, so let's move on to the next question in case no one has any doubt. So cells found around the stomata which regulate the pore size are called bundle sheath cells, lenticels, palisade cells, guard cells. So yes, the answer is guard cells. So what happens is when the stomata has to be opened, the guard cells take in a lot of potassium ions within themselves. So as the ion concentration inside the guard cell increases, the water uh, the water potential inside the guard cells decreases. So what happens is water moves from a higher potential to a lower potential. So water from the surrounding cells move into the guard cells, and because they swell up, the stomata opens. And when the stomata has to close, the K positive, the potassium ions go down in number. So when that happens, the water potential increases and the water moves out from the guard cells into the surrounding cells and the stomata closes. Okay. So the correct answer in this case is guard cells. Okay. So let's move on to the next question. Apical meristem is or are one primary meristem, two secondary meristem, three promeristem, four none of these. Yes. So as you're answering, the correct answer is primary meristem. So promeristem is the meristem cell or the premeristem cells that are present in the embryo itself, which is then converted to the primary meristem. So that is not the answer. Secondary meristem is what is responsible for the lateral growth of the plant, and that is also not the answer. So the apical meristem, which is responsible for the tallness of the plant, is a primary meristem. Okay, so the correct answer is primary meristem. So, in case there are no doubts, I will move on to the next question, which is which of the following is the precursor for auxin hormone? So, there are four options given to you: cytosine, tryptophan, methionine, indolacetic acid. Yes, the answer is in fact tryptophan. Indolacetic acid is the auxin itself, so that is not the correct answer. Methionine and cytosine are also not the correct answer. So there are three cycles through which tryptophan can be converted to indolacetic acid, indole 3 acetic acid, the IPA, the TAM, and the IAM pathways. Okay, so each pathway has different um, tryptophan will be converted to different compounds, which will again anyways finally end up forming indole acetic acid. Okay. So the correct answer in this case is tryptophan. So in case there are no doubts, I'll move on to the next question, which is which of the following pairs are haploid structures of angiosperms? Microspore and megaspore mother cell, anther and ovary, pollen grain and egg cell, and microsporocyte and ovule. Yes. So the correct answer is option number three, that is pollen grain and egg cells, which will have n pairs of DNA, and every other cell technically has two n pairs of DNA. So the correct answer is pollen grain and egg cell, which will fuse together to form a zygote. Alright. So let's move on to the next question. The dash generation is dominant in the bryophyte life cycle, and the dash generation is dominant in the seedless vacuolar plants. Gametophyte. The option one is gametophyte sporophyte. Option two is sporophyte gametophyte. Option three is gametophyte gametophyte, and option four is sporophyte sporophyte. So, what do you think the answer is? Any answers? I can't see any in the chat. That's why I'm asking, just in case there's a lag. Yes, you can unmute yourself and ask the question. Shruti and Vimal both have doubts. You guys have raised your hand. So, what is the doubt? Just tell me. You can write in the chat also in case you don't want to um, speak. Shruti and Vimal, I think. I think you guys had raised your hands. I hope you can hear me. Option one is the correct answer. Sure. Got it. Alright. Yes. So option one is in fact the correct answer. So in bryophytes, the dominant part of the life cycle is gametophytic. That means the cells have n number of DNA, and only a small portion of its life cycle is sporophytic, where it has 2n DNA. On the other hand, in seedless vascular plants, which are pteridophytes, they have the sporophytic cycle generation as their dominant phase, where they have 2n amount of DNA in their cells, and only for a small period of time will they have a gametophytic stage where the amount of DNA will be n. Okay, so if there are no doubts in this, you can. I'm hoping there are no doubts. Alright. Just in case there are, please write it in the chat box or unmute yourself, whichever is fine for you. Okay. So let's move on to the next question. Match the plant developmental process and the primary axis in which they occur. So you have activity of shoot apical meristem and root apical meristem. Second is activity of cambium in the secondary growth. Option 3 is development of upper and lower leaf surfaces, and option 4 is branching. And you have to match them with adaxial, abaxial, central peripheral, apical basal, and proximal distal. So that means these activities or the development process happen in a certain way, and out of these four, you have to choose which one they are. All right. So someone is saying option 2. Anyone thinks it's some other option? All right. All right, from the chat, I'm not seeing any other option. And yes, in fact, option 2 is correct. So that means 1 is 3, which is activity of shoot apical meristem and root apical meristem. So apical and basal. So it happens in this direction and in this direction, right? Second is activity of cambium in secondary growth, which is option 2, right? Central and peripheral. So it moves in this direction, cambium. That is what causes the thickness of the plant stem. Okay. Third is development of upper and lower leaf surfaces, which are actually different physiologically, right? And that is adaxial and abaxial. So the upper surface of the leaf is called adaxial and the lower surface of the leaf is called abaxial. Okay. So that is the correct option in this case. And branching is proximal and distal. Okay, so distal means as you move to the top of the plant or to the bottom of the plant from the junction of the root and shoot. Okay, so the further away you are, that is distal, the closer you are to the root shoot junction, that is proximal. Okay, any doubts in this? Just stop me anytime if you are facing any doubts. All right, so the correct answer is option 2, which matches all of these. All right. So I'm assuming there are no doubts as such. So let's move on to the next question. The short root gene is transcribed in dash, but the protein is transported to the dash. The options are vascular tissue, endodermis, endodermis cortex, QC vascular tissue, endodermis, and vascular tissue. Right. So people are writing the correct answer in the chat box. The answer is vascular tissue to endodermis. So if this is the vascular tissue, this protein will be transcribed here, okay, and through the plasmodesmata, it will be transported to the endodermis too. Okay, so the correct answer is option 1. The short root gene is transcribed in the vascular tissue, but the protein is transported to the endodermis. Okay, so that is the correct answer. Great. All right, now let's see the question. Next question. Alright, the ability of a single plant cell to give rise to the entire organism is called. There are four options: totipotency, cell division, de-differentiation, and none of these. What do you think the correct answer is? Yes. So, as people are saying in the chat, the correct answer is totipotency. So, plant cells have this, you know, what can it be called? Have the ability to from a single cell of a plant. It could be any plant cell, it could be from the leaf, roots, flower, any living plant cell has the ability to form an entire plant given the necessary conditions. Okay. Cell division is just division of one cell to give rise to two different or two separate cells. Okay, and de-differentiation is where a differentiated cell. So imagine this is a cell that is part of the vascular system of a plant. Okay, it is de-differentiated to form a cell that is just a plant cell, a callus, and nothing else. It is not a differentiated plant. It doesn't have a function in the entire system of the plant. It's just a plant cell and it's de-differentiated. Okay. So the correct answer is in fact totipotency and redifferentiation in this case, which is not in this given in the option, but redifferentiation will be when a callus is redifferentiated into a certain type of cell. Okay, it could be vascular tissue, it could be meristem, anything. Okay, so that is the correct answer. So let's move on to the next question. Which of the following is a chemical mutagen? DMSO, ethyl methane sulfonate, sodium chloride, and niacin. So you have to choose from these. So the answer is obviously ethyl methane sulfonate. DMSO is not a chemical mutagen. It is a very stable compound. Okay, and it is used to make solutions of a lot of compounds because it is inert in nature. Sodium chloride is just salt. That is also not a chemical mutagen. Does anyone know what niacin is? DMSO is,
It's a vitamin.
yes, it is vitamin B3. Niacin is a vitamin, so that is also not the correct answer. DMSO is, if you see a lot of procedures where you have to make extracts, right? So a lot of them will show you that DMSO is being used. So you don't know what the extract can interact with or form any chemical reactions with. So most of the time, not even water you use to make extract specifically. So you use DMSO. So what happens in that case is DMSO is completely inert. It mostly doesn't react with any chemicals. Okay, so in that case, you can form a extract with it. So DMSO is not the answer for those who thought that was the answer. The answer is ethyl methane sulfonate. Okay, all the other options are wrong. So, like you can see here, in what happens is when guanine reacts with EMS, it forms O6 ethyl guanine. Okay, and the change in the structure of this O6 ethyl guanine makes it form a bond with thymine. Okay, usually guanine will form a bond with cytosine, right? But in this case, it forms a bond with thymine. Okay, and by the time the cell's repair system comes to see this or to save this part of the DNA, what happens is it will assume that originally the thymine was there. So instead of this guanine being there, it becomes a AT bond in the repaired DNA. Okay, so that has changed. Okay, similarly, when thymine reacts with EMS, it forms O4-ethylthymine, which again will form a bond with guanine. Okay, so instead of a AT bond that you had, when the repair happens, okay, it will form a GC bond. Okay, so in that case, it is a chemical mutagen. It will change the bases in the DNA itself, and that will cause a change in the RNA and the protein being formed. Okay, and depending on if this is a coding sequence or if this is a very important amino acid in the function of an enzyme, it can cause a phenotypic effect to take place. Okay, so the correct answer is ethyl methane sulfonate. So let's move on to the next question. With reference to the TOS17 based transposon mutagenesis, which of the following statement is false? Okay. So the options are TOS17 is a kind of retrotransposon, TOS17 is activated by tissue culture, TOS17 is inactivated in regenerated plants, and TOS17 is inactivated by tissue culture. So what is false in this? All right. So a lot of you are saying the answer is 4. And yes, the answer is in fact 4. So TOS17 is a kind of retrotransposon. So what a retrotransposon is, it is a portion of the DNA, all right? So what happens is it will be transcribed into RNA, then translated into protein, or sorry, we transcribed into RNA, translated into protein, and this RNA that is there, it will be reverse transcripted, okay? So that RNA is converted to DNA, and this DNA will be reintegrated into a different part of the DNA itself. Okay? So TOS17 is a retrotransposon. It is activated by tissue culture, which is true. So in mature plants, this is not going to jump genes. Okay, that is, it won't get integrated into different areas. But once you convert a cell from the mature plant to a callus, it will activate this gene, okay, TOS17. So it is activated by tissue culture, which is correct. I'm sorry, I couldn't hear you. The connection might not be great, so can you type it out in the chat in case I'm still not able to hear you? The person who had just unmuted themselves, did you have a doubt? Like, are you typing? I can't see anything in the chat itself. All right. Since I'm not seeing anything in the chat, I'll assume there wasn't a doubt. All right. So yeah. So what happens in this retrotransposon is the DNA is converted to RNA, and that RNA is reverse transcripted to DNA, which is then integrated into a new area of the genome. Okay. So it is inactivated in regenerated plants, which is also true. So once a plant starts growing and it becomes mature, TOS17 is inactivated. So the false option is that it is inactivated by tissue culture, which is not all right. So uh, from this you could have figured out either this has to be wrong because they are the opposites of each other, right? So great for everyone who got the answer. So let's move on to the next question. The question is a molecular marker with which one is able to differentiate between a homozygous and a heterozygous condition is called a epigenetic marker, dominant marker, codominant marker, and none of these. Alright, I see a few answers in the chat. Any other answer that you think is correct? Alright. So the consensus is option number three, right? A codominant marker. So yes, that is the correct answer. A codominant marker is the correct answer. So what you see here is a gel in which a dominant marker is used, and this is a gel where a codominant marker is used. All right. So that marker in the dominant marker where the dominant marker is used, only where the organism has the dominant gene, which is in this case capital B, is present. Can you see that? All right. And in this case, you can see all of them, right? So you can see a dominant marker, a dominant organism where it has AA, All right. A recessive one, which in which it has small a, and a heterozygous one also, which has capital A small a. Okay. So the correct answer is codominant marker. Okay. A dominant marker will only help you find whichever has the dominant gene in it. Okay. So that is not the correct answer in this case. So let's move on to the next question, and this is the technically the last question for today as such. So the question is, a molecular marker co-segregates with the mutant phenotype of your interest. Okay. Which of the following statement is false regarding the observation? Option 1 is
Option 3, ma'am.
option 4. All right.
3, ma'am.
3. All right. So you're saying option 3. So let me just read out the options. The molecular marker and the mutations are present on the same chromosome. The molecular marker and the mutation are present very close to each other. The molecular marker and the mutation are present on different chromosomes. And the molecular marker and the mutation show genetic linkage. So yes, you are correct. The answer is in fact option 3. So if they were on different chromosomes, you will not see co-segregation. So just because the molecular marker is there in the cell, it does not mean that the mutant phenotype will also be there in the cell. Okay. During recombination, if they were on different chromosomes, definitely you would get recombinant products in which either has only the molecular marker or only the mutant phenotype. Okay, but since they are linked like this, like you can see here, they're very close to each other. So what happens is, no matter even if recombination takes place, okay, you keep getting both the genes together. So the molecular marker and the mutant genotype keep um, you know uh, going with each other. Okay, so they show linkage because they are close by to each other and all obviously present on the same chromosome. So that is the correct answer. So for those who joined late, I'll start again. All right. So that was the last question technically for today. Um, and those who have seen all the questions who were there from the start, you guys can leave the meeting unless you have any doubts. So if you have any doubts with regards to the course or anything else, you can ask me. If it is regarding the assignment for this week, we will discuss this after the due date of the assignment is over. Okay. So anything apart from that, you can ask me if you have any doubts or if you have any suggestions, you can tell me that also. All right. So those who were there from the start, you guys can, if you want, leave. I'll be repeating the same questions again. Okay. And those who joined late, you can stay back. I'll just restart the questions. Okay. All right. So I'm starting again. These are the questions of the 0 week assignment. In case you had any doubts, we'll just be discussing them. Okay. So the first question is, which of the following is the male reproductive organ in a flowering plant? So there are four options given to you: the calyx, the gynoecium, androecium, and corolla. So those who joined late, what do you think the correct answer is? Three. Yes. So yes, the answer is androecium. Gynoecium is the female reproductive organ. The calyx is the sepal, all right, the leafy part of the flower, and the corolla is the petal, all right. So the correct answer is androecium. Okay, androecium is the male reproductive organ in the flowering plant, which is made of the anther and the filament. Okay, and together they form the stamen, and that is the androecium. All right. So that is the correct answer. Let's move on to the next question. The next question is that the energy currency for any living cell is RNA, DNA, ATP, and AMP. So which is the energy currency that all living cells use? Yes. So the answer is ATP. adenosine triphosphate is the correct answer. So as you can see in the diagram, this is ATP, adenosine high triphosphate. So the bond between these phosphate molecules is a high energy bond. So it takes a lot of energy to break them, and once you break them, right, you release a lot of energy. Okay, so an enzyme called ATPase will break this bond. The inorganic phosphate will be released, and because of this reaction, you will have free energy released, which can be used for other processes. Okay, so usually this is combined with another process that requires energy. Okay, so the correct answer is ATP. AMP, adenosine monophosphate, is where you don't have these two phosphates. Okay, so now that you don't have these two phosphates, there is no high energy bond to break, so it is not a energy currency. RNA and DNA are nucleic acids. Okay, so they are also not a energy currency. So let's move on to the next question. The question is, cells found around the stomata which regulate the pore size is called bundle sheath cells, lenticels, palisade cells, and guard cells. So what do you think is the correct answer? Option 4, guard cells. Yes, the answer is in fact option 4, guard cells. So guard cells are what you see here. Alright. So these are guard cells, and how they work is when the stomata has to be opened, that is the pore size has to be changed in this case. Okay. So when the stomata has to be opened, what happens is the guard cells have an increase in potassium ions. Okay. When the potassium ion increases, the ion concentration inside the guard cell increases. Okay. And the water potential decreases. So once the water potential decreases, what happens is water moves from a area of higher potential to a area of lower potential. So from the surrounding cells, the water moves inside the guard cells, okay, and that causes the guard cells to swell up. And when that swelling happens, the stomata opens, okay, because the guard cells take this shape. And when the stomata has to be closed, the potassium ions in the guard cells go down, okay, and the water potential increases. So the water H2O potential increases, so the water moves out from the guard cells to the surrounding cells. And because the guard cells shrink, the stomata also closes down. Okay. So the correct answer in this case is guard cells. So let's move on to the next question. The question is, apical meristem are option 1, primary meristem, option 2, secondary meristem, option 3, promeristem, and option 4, none of these. So what do you think the correct answer is? Option 1. Yes. So the answer is in fact option 1. Apical meristem is the primary meristem, and apical meristem and root meristem are meristems which are primary meristems, and they help in the growth of the plant, that is the tallness of the plant or the length of the root. Okay. So they are primary meristems. Secondary meristems will be lateral meristems which help in the outward growth of the plant, that is the increase in thickness of the plant. And promeristem is what was there before the primary meristem. So promeristem is converted to the primary meristem from the embryo. Okay, and obviously none of the above are is not the correct answer. Okay, so let's move on to the next question. The question is, which of the following is the precursor for auxin hormone? Cytosine, tryptophan, methionine, and indole acid. So, what do you think the correct answer is? Option 2. Yes. So, the correct answer is tryptophan. So, tryptophan is the precursor, and along three pathways, okay, it can form indole acid, which is auxin. So, indole acid is auxin itself, not the precursor. Okay. So, this is not the correct answer. Methionine is also not the correct answer. Cytosine is also not the correct answer. The correct answer is tryptophan. So there are three pathways: the IPA pathway that you can see here, where tryptophan is converted to indole 3 pyruvic acid, then indole 3 acetaldehyde, and then finally indole 3 acetic acid. Another pathway is the TAM pathway, where tryptophan is converted to tryptamine, which is converted to indole 3 acetaldehyde, which is converted to indole 3 acetic acid, and the IAN pathway, where tryptophan is converted to indole 3 acetyldoxamine, and which is converted to indole 3 acetonitrile, which is then converted to indole 3 acetic acid. Okay? So along these three pathways, the plant can produce auxin hormone because it has the precursor tryptophan. Okay? So that's for this question. Let's move on to the next question. Which of the following pairs are haploid structures of angiosperm? Microspore and megaspore mother cell, anther and ovary, pollen grains and egg cell, microsporocyte and ovule. So, what do you think the correct answer is? Anyone? You can take a guess also already if you don't know the answer. This is a problem solving question. Right? Yes, the answer is C or 3. Yes, it is pollen grains and egg cells. So anther and ovary, even though they are the reproductive organs, they have 2n number of DNA. Okay? And the cells within the anther and the ovary will be converted to pollen grains and egg cells, which will be n in number. And the pollen grain and egg cell will fuse together to form the zygote, which will again be 2n. Okay. So the correct answer, that is haploid. Haploid is cells which have one set of DNA, and diploids are cells which have two sets of the DNA. So of the following pairs are haploid structures of angiosperms. Angiosperms are plants which are flowering plants, right? So that means their reproductive organs are flowers. Okay. So in that case, the haploid structures or structures which have only one set of DNA are pollen grain and egg cell. Alright, let's move on to the next question. The question is, the dash generation is dominant in the bryophyte life cycle, and the dash generation is dominant in the seedless vascular plants. The options are: option 1 gametophyte and sporophyte, option 2 is sporophyte and gametophyte, option 3 is gametophyte and gametophyte, and option 4 is sporophyte and sporophyte. So, what do you think the correct answer is? I can't see any answers in the chat. Do you guys have a doubt in this question? Just take a guess, that's fine. Option B, sporophyte, gametophyte. Alright, anyone thinks it's something else? Alright, since I see no other option. Um, the correct answer is option 1, that is gametophyte and sporophyte. So the gametophyte generation is dominant in the bryophyte life cycle. So this is the bryophyte life cycle. These are the gametophytes. So there is a female gametophyte and a male gametophyte. Alright. which will form these archegonial heads, which is the female reproductive organ, and the antheridial head, which is the male reproductive organ, and they will form uh, gametes, which will fuse together to form the spore. Okay, and that spore will germinate into again the archegonium and endometrium, right? Which will give rise to your gametophytic stage again. So, for the maximum life cycle, the plants or the bryophytes in this case are gametophytes, and only for a small duration are the sporophytes. Okay, so um, yeah, so this is pteridophytic life cycle. So seedless vascular plants are pteridophytes. They are also known as pteridophytes. Okay, so for most of their life, they are sporophytic in nature. Okay, that means they have diploid uh, DNA content, and only for a small period of time will they be haploid. Okay, so they will have a small sporophytic stage. Okay, where they will form the gametes or the embryo required for the formation of a new uh, pteridophyte. Okay, or fern in this case. All right. I hope there was no doubt in this question. In case you have any doubts, just write it in the chat. Okay, I'll just go over that again. So let's move on to the next question. Match the plant developmental process and the primary axis in which they occur. Okay. So the developmental process is activity of shoot apical meristem and root apical meristem. Then is activity of cambium in secondary growth. Then it is development of upper and lower leaf surface areas, and it is branching. Okay. There are four options given to you. Okay. There are four options given to you, and you have to choose from these four options where each of these axes are. Okay, these development process follow which axis. Okay, so take your time and maybe if you don't know, try to guess what the correct answer will be. Anyone has any guesses of what the correct answer is? Option two. Yes, that is correct. The correct answer is in fact option two. So the first one was activity of shoot apical meristem and root apical meristem. So as you know, if this is the plant, right, and these are the roots, a taproot system maybe, let's imagine. Okay, there's a shoot apical meristem and there's a root apical meristem. Okay, so the shoot apical meristem is what is responsible for the growth of the plant upward, that is the increase in the length of the plant, and the root apical meristem is responsible for the growth of the root, that is the increase in length of the root more towards the downside. Okay, so the activity of SAM and RAM is apical and basal. Okay, so in the direction of top or bottom. So that is apical and basal, right? So second is activity of cambium in secondary growth. Secondary growth is when the thickness of the stem increases. So that means from the center to the periphery, right? The growth increases. So this is the correct answer for that. Okay. The third one was development of upper and lower leaf surfaces. So the upper and lower leaf surfaces are different physiologically itself, okay? And that happens in the axis called adaxial and abaxial. So any leaf top surface is called adaxial, okay? and its bottom surface is called abaxial. Okay, so the correct answer for that is uh, adaxial and abaxial. And finally, branching. The correct answer is proximal and distal. So the closer you are to the root shoot junction, it is proximal. The farther away you are, it is distal. Okay, in both sides, in both directions. Okay, so branching is proximal and distal in nature. Okay, so I hope that was easy for you to understand. Let's move on to the next question. The short root gene is transcribed in dash, but the protein is transported to the dash. There are four options given to you. Option 1, vascular tissue endodermis. Option 2, endodermis cortex. Option 3, QC and vascular tissue. Option 4, endodermis and vascular tissue. So what do you think the correct answer is? Madhyam C has raised hand. Do you have a doubt? You can write it down in the chat box or unmute yourself, but the connection might not be clear, so I might not be able to hear properly. But you can try. Nope, not a problem. Do you want to answer the question or do you have a doubt?
Fourth one option, ma'am.
You're saying fourth is the answer? All right. Or first one. I didn't hear you properly. Which did you say is the answer? All right. So, since I could hear you, the answer is in fact one. So, the short root gene is transcribed, that is, the gene is transcribed into RNA from DNA in the vascular tissue. But the protein is transported to the endodermis, okay, to the plasmodesmata, that is in this direction. So the gene is transcribed somewhere and translated somewhere else completely. Okay. So that is it for this question. Unless there are any doubts, I'll move on to the next one. So the next question is the ability of a single plant cell to give rise to the entire organism is called. There are four options given to you. Option 1 is totipotency, option 2 is cell division, option 3 is de-differentiation, and option D is none of these.
First, ma'am.
Yeah, so the correct answer is in fact totipotency. So plant cells are totipotent in the sense that no matter what cell you take from it could be a leaf cell, okay, it could be a flower, it could be ovules, it could be from the stem, the root, any of these cells you take, under the proper conditions, these cells can be de-differentiated and then re-differentiated to form a completely new entire plant. Okay, so that is the totipotency of the plant cells. Cell division is just the division of the cells, so that is not ability as such of plant cells. Okay, de-differentiation is when a cell has already divided and formed or differentiated for a particular purpose. So the cell has already become a vascular cell, all right, or some other cell, and it is de-differentiating back into a stem cell sort of phase. Okay, so this is differentiated and this is de-differentiated. So that is what happens when you form a callus from mature cells. Okay, and this can be re-differentiated to form a new type of cell, or in because they're totipotent, a completely new plant. Okay, and none of these is not also the correct answer. So let's move on to the next question. Which one of the following is a chemical mutagen? DMSO, ethyl methane sulfonate, sodium chloride, and niacin. So what do you think the correct answer is? Option 2. So yes, the answer is in fact option 2. Ethyl methane sulfoate is ethyl methane sulfonate is the correct answer. DMSO is an inert substance. It is usually used for forming extracts or in experiments where you don't want whatever is your compound of interest to react with anything. It's very inert. Okay? Sodium chloride is salt and niacin is a vitamin. Vitamin B3 is niacin. The correct answer is ethyl methane sulfonate. So as you can see here, when guanine reacts with ethyl methane sulfonate, it forms O6 ethyl guanine. Okay. So what happens is the structure of that changes the guanine so much that instead of forming a GC bond like it normally will, it forms a GT bond. Okay, and when the cell's repair system repairs this, it will recognize this ethyl guanine to be incorrect. Okay, like not meant to be in the DNA, but it will not know what the original sequence was. So what it will do is it will see that T is here, and it will convert that to AT base. Okay, so instead of the original GC that was there, it will become AT. Okay. Similarly, in the case of thymine, when it reacts with ethyl methane sulfonate, it forms O4 ethyl thymine. Okay, and this instead of forming a bond with T like it would normally an AT, it forms a TG bond. Okay, again when this is repaired, what happens is this T will be converted to C because the cell will perceive that the G was there originally. Okay, so a AT will be converted to GC, and depending on if it is in a coding region and if it codes for a important amino acid in an enzyme or a protein, it can lead to phenotypic changes. Okay, that can be deleterious in effect. Okay, so the correct answer is ethyl methane sulfonate. Let's move on to the next question. The question is with reference to the TOS17 based transposon mutagenesis. Which of the following statement is false? So option 1 is TOS17 is a kind of retrotransposon. TOS17 is activated by tissue culture. TOS17 is inactivated in regenerated plants, and TOS17 is inactivated by tissue culture. So what do you think the correct answer is? Get activated which stage? In this case, what is the stage that you're referring to? Just for clarity, like what are the stages that you're referring to? All right. So, um, in this case, I will tell you what the false answer is. Okay, of tissue culture, like callus culture. Okay, got it. So yes, so that would give away the answer in this case. Okay. So the correct answer is it is inactivated by tissue culture. Okay. So in this case, it is not mentioned specifically in the question itself, but what this means is the callus stage itself. Okay, the tissue culture. So option one, the TOS17 is a kind of retrotransposon is correct. So what a retrotransposon is, that this is the retrotransposon gene in the DNA. Okay, it will be transcribed and translated. Okay, to form something called a ribonucleoprotein complex. Okay, and this will reverse transcriptase the RNA to DNA, which will then get integrated into a different portion of the genome. Okay, so this is how it is jumping in the genome itself. This gene does not work in a mature plant. Okay, so option one was technically correct. It is activated by tissue engineering or tissue culture, means that when you form a callus from the mature plants, like you were saying, this gets activated. Okay, so that is another correct answer. And the option number three, that is TOS17, is inactivated in regenerated plants. That means when this callus is redifferentiated into a full plant, this gene will be inactivated. Okay. So 1, 2, 3 is correct. The only option that is false is option number 4. That is TOS17 is inactivated by tissue culture, which is the exact opposite of option number 2. And since option number 2 is correct, option number 4 is wrong. Okay. So in case there are no doubts in this question, I'll move on. If there are doubts, just please write it in the chat. Okay. So this question is a molecular marker with which one is able to differentiate between a homozygous and a heterozygous condition. Epigenetic marker, dominant marker, codominant marker, and none of these. So what do you think the correct answer is? Option 3, codominant marker. Yes, the correct answer is option 3, codominant marker. So as you can see in this diagram, one is autoradiogram of a dominant marker and another is that of a codominant marker. Okay. So like you can see here, the marker is only for the dominant allele, right? So only organisms which have the dominant allele will be shown here. Okay. So that is the capital B and the capital V small b. Okay. And in the codominant marker, you can see all three. All right. Capital A, small a, and capital A, small a. Okay. So dominant marker will only tell you whether the dominant gene is present or not. Okay. It will not tell you if it is homozygous or heterozygous. Okay. Um, so, any doubts in this question? All right. I'm assuming no doubts are there. So, this is the last question for today. The question is, a molecular marker co-segregates with the mutant phenotype of your interest. Which of the following statement is false regarding this observation? Option 1 is the molecular marker and the mutation are present on the same chromosome. Option 2 is the molecular marker and the mutation are present very close to each other. Option 3 is the molecular marker and the mutation are present on different chromosomes. And option 4 is the molecular marker and the mutation show genetic linkage. So you have to find out which is false regarding the observation. So what do you think the correct answer is? Alright, I don't see any answers in the chat. So let me tell you what the correct answer is. You have to find out what is false in this, right? So let's check one by one. The first one is the molecular marker and the mutation are present on the same chromosome. So when they're saying that a molecular marker co-segregates with the mutant phenotype of your interest, that means no matter what cell you take, if the molecular marker is present, then the mutant phenotype is also present. Okay, so they are not separated at all. So that means they must be on the same chromosome. Okay, otherwise, if they were on different chromosomes, the chances of you having a cell with only one of them will be high. Okay, so this is correct. The second one is the molecular marker and the mutation are present very close to each other, which is also correct. So like you can see in this case, when two genes are close to each other, it becomes very difficult for them to move separately, even when you have recombination. Okay. So even when recombination happens, since they are very close to each other, chances are that they move together, okay, even upon recombination. So since you are seeing the molecular marker co-segregate with the mutant phenotype continuously, that means the second one is also correct. Option C is the molecular marker and the mutation are present on different chromosomes. So, since option 1 was correct and this is the opposite of the option 1, this is wrong. Okay, let's see what the fourth one is. The molecular marker and the mutation show genetic linkage. So that is also correct since they move together. They and because of their close nature, due to the distance between them, they show genetic linkage, which is also correct. So the incorrect option or the false option in this case is option number 3, okay, which is the answer for this. So that's all the questions that are there for today. Alright. So in case you have any doubts, you can ask me. If you don't have any doubts, you can leave the meeting and at the same time next week we'll have the problem solving session for week 2 and I think till then the assignment due date is not finished. So maybe weekly we can discuss the first assignment and so on. Alright. Thank you so much for joining. You guys can leave if you don't have any doubts or any questions for me. Those who have can stay back. Do the people who have stayed back have any questions? I'm sorry I couldn't hear you. Did you say something?
Yeah, ma'am. I'm ready.
Yeah, what did you say? Do you have a doubt?
No, ma'am.
All right. Then yeah, if you don't have a doubt, you can leave the meeting. Not a problem.
Okay, ma'am.
Sure. Thank you. Hi, the person who just joined right now. The class for today is technically over. We'll be uploading these onto YouTube. You can watch the video version of that there. Do you have any specific doubt?
No, ma'am.
All right. If you don't have any doubt, anything else you want to see the questions that were taken today?
Yes, ma'am.
Uh, Wait, Vimal, you have already seen them, right? I was talking to the person who joined just now. What's your name? Just let me see. Karthi, MG, right? You just joined right now, right? So the class for today is over as such. We've taken all the questions. Um, what do you want to do? You will watch the video or do you want to go over the questions? Video link. All right. I don't have the link right now. I'll have to um, finish this first and then upload it to YouTube. And then the NPTEL team will send or uh, sub- give you the link. All right. It'll be there somewhere in the page itself. So I think video recordings will be shared with you. Not to worry. All right. So if there are no other doubts, I'm going to close this meeting. Okay, thank you so much for being here. Let's see you next week. That's
Thank you so much, ma'am.
all right. Thank you so much. Uh, Sanjana, the questions or the meet for today is technically finished. Like we've gone over the question set twice. So, what do you want to do? You want to see the questions, or there'll be a link provided to you guys later of today's class on YouTube. So, what do you want to do? You just want to go through the questions and try to see if you can solve it. All right, so I'm going to close this meet. See you all next week.